good. So thanks, Alex, for the invitation, and thanks for your group for all the administrative support in the back just to get this all set up. So that was great. Thanks for setting this all up. And I've been told I have about 30 minutes to speak, so I'll try to keep to time here, and I'll leave some time for discussion as well too. So we are still in the process of developing these preclinical reporting guidelines. I'll give you a little bit about the rationale and the process we've gone through so far. And I will mention that there's still room to actually provide input there. We have, I don't get into it, but some of the criteria we're looking for in terms of if you want to join for the next round of voting, basically. So with that being said, my objectives for today are to provide a brief introduction to meta-research and systematic reviews. It sounds like we got some more junior folks here. I read from the, what they were hoping to see in terms of an introduction to systematic reviews. Unfortunately, I'm not going to give much of that, but I'll give you a bit at least of my perspective on meta-research and systematic reviews. Current reporting and preclinical systematic reviews. So there's a study that I think several of you have seen before because we've discussed it in terms of reporting in preclinical systematic reviews to date. And then finally, a little bit about PRISMA-Pre and its development. So in other words, as a reporting tool for preclinical systematic reviews. Let's just jump right into it in terms of meta-research. Ultimately, I think meta-research can help us. There's other names for meta-research or research on research or the science of science. So this is the number of articles published per year in just one small field. And you can see that there's a steep increase occurring in the early 2000s, largely with the advent of mega journals and also the advent of publishing online only. We saw skyrocketing of articles. Now, to keep on top of all of that, I think we need to evaluate and improve our research practices as well too, and meta-research can certainly help us do that. In fact, I think if meta-research is used effectively, it can help us improve science, it can help us improve medicine. And I didn't really introduce myself there. Alex provided a bit of one, but I am an anesthesiologist. I'm a clinician by background and still practicing anesthesia. So ultimately, I think always about the health of our patients and how even what we're doing in terms of in the lab or in terms of preclinical systematic reviews, always downstream, we think about our ultimate end user, which is our patient. So, it doesn't matter what sort of research you're in. If you're doing anything biomedical, I would suggest that the ultimate end user for your work is actually patients and all of us. So, so ultimately, I think meta-research can help us meet those goals and improve science. In terms of systematic reviews, this is a little cartoon I'm sure many of you are familiar with from Cochrane, basically showing the different steps of systematic reviews. But I did want to present the PRISMA 2020 statements definition of systematic reviews, which is that it's a review that uses explicit systematic methods to collate and synthesize findings of studies that address a clearly formulated question. Also, in terms of definitions, I also wanted to define right up front what I mean by preclinical. So it's really funny, when I started in this field, I just used the word preclinical and I had my definition in my mind, which is in vivo animal experiments. But now, having done this for almost a decade, I recognize that that word preclinical means many things to many different people. So I always try to define that word up front, just so we're all on the same page as to what we're talking about. In terms of where systematic reviews fit, I really like this cartoon. We made this, but it's actually adapted from an article that Kim Weaver, which many of you know, she was a co-author on it. But I really like the way they sort of laid it all out. But it's really based on what we do clinically. So in clinical trials, we have a lot of single center studies, and often from those single center studies, we'll do systematic reviews and meta-analysis, and that'll help us justify the conduct of a large confirmatory study. So in this case, usually multi-center RCT is usually what we're thinking about when we're thinking about a confirmatory clinical study. And then, if there are findings that are of use to the community, then we go into stage 4, which is more implementation and de-implementation of these practices based on these findings. And I should probably have another little box here around systematic reviews of these confirmatory studies, because that's often what Cochrane does, for instance, right? In some of their more practice-changing systematic reviews, where they're looking at more later stage evidence and what the efficacy or safety might be of a certain intervention. Now let's flip over to laboratory studies and what we do in the lab. Well, I think the systematic reviews still have a role to play between the stage 1 and stage 3. So basically you have all these single lab exploratory studies being conducted. And from those, we can often conduct systematic reviews and meta-analyses that can either identify a knowledge gap that we can go back to stage 1 and fill, or it could also help us justify potentially a confirmatory study or some form of confirmatory study. And for those of you that are largely basic scientists, you'll know that this whole terminology around confirmatory studies is still relatively new to the field. But I'll show you a couple of examples of how we use that. However, once you get to the stage 3, then often these sort of more confirmatory studies, or quite often just a bunch of single lab studies where we've done systematic review, can help justify the conduct of a early phase clinical trial. That's certainly what our group has done in multiple instances. Throughout all of this, I just want to point out that our group and many others were very pro stakeholder engagement throughout the entire process. So, we even bring patient partners, patient engagement, even at this level, at the very top here. But of course, even we're doing these early experiments certainly in our research institute, we like to have clinicians who'd be running trials downstream on board with those teams as well too, which only strengthens them. So in terms of our group's use of systematic reviews, this is one example, looking at extracellular vesicles from a particular cell type and looking at respiratory diseases. What we found from this particular systematic review was that there was this knowledge gap. No one had actually looked at the biodistribution of these extracellular vesicles in lung injury. Lung injury is one animal model that we're quite interested in. So we were able to identify this knowledge gap, and then that led to a study where we did exactly that to fill the knowledge gap. We looked at the biodistribution of these extracellular vesicles in acute lung injury. So I think a really nice example of how a systematic review can identify a knowledge gap, and if you have the right people around the table, you can actually fill that knowledge gap pretty swiftly as well too. So in addition, this was just hot off the press, the final proof we just sent in, but you can see it online if you have access to the American Journal of Physiology, Lung and Cellular section. We basically conducted this systematic review assessing biological sex, again, in a lung injury model. We identified a few studies, but again, we actually demonstrated this large knowledge gap on just such a fundamental question. What is the difference between male and female animals in one of the most popular, if not the most popular lung injury model that's used around the world, which is this lipopolysaccharide-induced lung injury. So what this is doing is actually helping justify and set up for a multi-laboratory study that we'll be running in the next few months, where 4 different labs will be addressing this specific question, inducing the disease, and then looking at differences between male and female mice. So I think some pretty exciting uses of systematic reviews that fits in exactly to that translational pathway I was providing a little bit of an overview in the previous slide. So let's think a little bit more about systematic reviews and preclinical systematic reviews. In terms of what's out there, well, we conducted this study now published a couple of years ago, where we looked at the epidemiology and the reporting characteristics of preclinical systematic reviews. So what are people doing? How are they reporting it? I've got a QR code here, and I understand this is being recorded and posted online as well too. So feel free to go back to the slide and do the QR code. You can just look up Hunneford and PLOS biology, and you'll find this paper pretty quickly. The objective of this particular paper was to assess the prevalence and the reporting of preclinical systematic reviews. And the methods that we use, well, we try to be systematic, of course. We're here evaluating how other people have conducted systematic reviews and meta-research. So we try to be rather systematic and rigorous ourselves. We performed a systematic search with the help of an information specialist, our medical librarian here at the Ottawa Hospital. Then we did an assessment of reporting. I don't get into all the details, but all the usual that you would expect from a well-conducted meta-research or systematic review study, we tried to abide by. Basically, we had piloted checklists ahead of time. We ensured that there was good inter-rater concordance as well prior to completing our data extraction. And we, of course, pre-registered our study. It was on OSF, where you could find a registration for this particular evaluation of systematic review reporting. Just briefly, we had about 2,400 records that we had to screen through. Ultimately, at the end, we found 442 systematic reviews of in vivo preclinical studies. So again, when I'm talking about preclinical studies or preclinical systematic reviews, these were largely in vivo or systematic reviews of in vivo work, looking at an intervention as well too. These were published from 2015 to 2018. We've updated a little bit of this. I don't have that data here, but really no differences. And that's not surprising. I'll show you some of the results, obviously. Not surprising because between when we actually extracted this data set and present day, there's been very little intervention in terms of trying to address reporting within preclinical systematic reviews. What was very interesting, though, was that in terms of our full text exclusion, 73 reviews were actually mislabeled as systematic reviews. So they said, oh, it's a preclinical systematic review, it's a systematic review of animal studies. They were not. And we were actually quite generous, I would say, in our criteria. They had to have a focused research question and a systematic search. Like basically those are two requirements. There's other ways to define what a systematic review was, but if they did not have those two things, we did not include it in this data set, basically. So, and this is not unique to preclinical systematic reviews. Doing similar work in the clinical systematic review world, there's certainly plenty of clinical systematic reviews that are not systematic reviews. So just keep that in mind. In terms of the number of systematic reviews that we found, or preclinical systematic reviews, this is actually data from a study that actually inspired this current study, published in PLOS One a few years before ours. And you can see here there's a steady increase in what they found. They had very similar definitions as to what they were going to include or exclude. You can see the y-axis is a total of 35 here on the top, so not that many. Continuing on that graph, this is not exactly what's in the paper because we've updated it, so there's a bit more updated data here. But what you can see here, first of all, our y-axis is quite different. We're talking hundreds here rather than the top of the scale being 35. And you can see that there's this nice steady increase, not surprising given the increase in overall medical literature, but also I think it is, even when you normalize it to the total number of articles published per year, we've done that. Basically, there's no difference. So you can see that there is proportionally a larger increase in preclinical systematic reviews being published year over year. So exciting. It means that there's more people interested in the same things that we are. And I think that opens up the room for some guidance as well, too. In terms of where these studies are coming from, and this is going back to just 2018, but you can see there's some hotspots here in terms of where absolute numbers of these preclinical systematic reviews are being published. The United States, Brazil, China. But what's really nice here is you can see representation from a large number of countries all across the world. So this is truly an international sort of effort, and there are these preclinical systematic reviews being pursued in a number of different parts. Nice little graphic that we just designed. I thought was kind of fun, but just looking at the numbers of studies that actually were reported on these types of animals. So not surprisingly, rat, mouse, rabbit, right at the top of this pyramid in terms of absolute numbers of preclinical systematic reviews reporting on studies that were conducted in rats, mice, and rabbits, rats, and mice, excuse me. And then not surprisingly, looking at larger animals, fewer of those studies being done. And I certainly being a preclinical researcher myself, probably reflective of the costs that are associated with these large animals. Reporting assessment, I won't bore you with all the details, but I'll give you a few takeaways from what we found. First of all, the introduction was the most completely reported section. 2/3 of the reviews reported on all the 3 items that we assessed. And I should mention, in terms of what we assessed, we basically modified the PRISMA checklist, the one that exists for clinical systematic reviews, and we used that for our assessment. The 3 items, just so you're aware, for the introduction were objective rationale and a focused research question. Objective and rationale were given most frequently. The focused research question, or sort of PICO question, which we'll circle back to later on for those of you that are less familiar with it, that was actually one of the items that was less frequently reported. Reporting was the lowest in the methods section, followed by the results, which is very disappointing, right? So if you want to conduct a study and you want to replicate it, basically you're relying on the methods that are being reported. So unfortunately, in terms of all the different sections, introduction, methods, your results and conclusion, the methods was the most poorly reported section overall. For instance, only 20% reported an a priori protocol being posted anywhere. Less than half of the reviews reported a risk of bias assessment. So these are, I think, highly problematic within the systematic review world. However, obviously, from our side, offers opportunities for improvement as well, too. Reporting overall, though, which was nice, has improved in some areas, because I did show you that previous assessment that was published in PLOS One in 2014. And our assessment or review demonstrated there was actually a quantitative increase or improvement in reporting across the board, though. However, that being said, inconsistent reporting across manuscript sections was demonstrated to be an issue. Certainly, again, an exciting opportunity for us to fill. So what's our solution? Well, guidelines. And for those of you that are maybe not familiar, Equator is the clearinghouse in the world globally for reporting guidelines. When you go onto their webpage, this is just a screenshot of it. You can see a number, just right on top, you can see what they recommend for randomized controlled trials, consort, observational studies, strobe, getting to animal work. So animal preclinical studies is ARRIVE. And of course, going back up here to near the top, systematic reviews, they recommend PRISMA. And I think most of you are familiar with PRISMA, but basically it's a checklist. It was updated in 2020, and it's a guideline for reporting systematic reviews, largely centered around clinical systematic reviews. And just so you understand the history of all this, it was really set up to look at interventions as well, too. So when you're doing systematic reviews of observational studies, there are additional elements that you should consider, for instance, because PRISMA, the statement in and of itself, is really looking and designed specifically for RCTs and interventional human studies. Within this, you'll see this checklist. Again, this is part of the checklist only, but it's split up by the sections you'll be reporting within your study. So title, abstract, introduction, etc. And in terms of what it does, it's IDing essential items and approach completeness and transparency in your reporting. History-wise, again, it was actually first put together in 2009, cited about 120,000 times. It has an explanation elaboration, and that's been cited more than 35,000 times. I just looked this morning to look at what the parent PRISMA statement was, how often it's been cited, and it's been cited already 53,000 times. So not surprising, just given the fact, again, that the number of clinical systematic reviews has dramatically increased as well, too. So PRISMA acts as a learning resource for best practices, and it's improved reporting in clinical systematic reviews. And this has objectively been shown in several studies at this point as well, too. So the publication and the implementation and the adoption, I should say, of PRISMA has actually quantitatively improved reporting in clinical systematic reviews. So of course, we're trying to think, well, or we're hoping that similar results would occur from PRISMA-Preclinical, excuse me. I want to mention one thing here, though, before I move on, which is that PRISMA truly is a reporting guideline. It is not a conduct guideline. So when I'm a peer reviewer for systematic reviews and I see a line in the method saying the PRISMA guidelines were used to conduct the systematic review, it already raises a red flag for me, because it demonstrates to me that potentially these authors aren't aware of what PRISMA actually is. It's a reporting guideline, it's not a conduct guideline. But that being said, as I've mentioned in the last sentence here, it does actually help set up for best practices as well, right? So if you are reporting all these items, then hopefully you've conducted those well, and you've conducted all the things that they are suggesting in terms of actually completing a clinical systematic review. In terms of conduct, if you are looking for best resources, I would suggest, obviously, the Cochrane Handbook. If there's a Bible for clinical systematic reviews, it is the Cochrane Handbook. And as well, JBI has some great resources as well, too. And I know from your center in Berlin, you have lots of resources set up as well, too, to guide people through the preclinical systematic review process and the systematic review process. So in terms of a need for preclinical extension, again, there's no guideline for reporting systematic reviews of preclinical studies. And many of us in this field who conduct preclinical systematic reviews feel that PRISMA, the parent statement, and even the latest 2020 version is insufficient. And there are some unique issues that we need to consider that I think make that statement on the top true. So number one, reporting. There's a variety of methods and species used to induce disease. It's not just humans, right? So we need to think about reporting those differences. There's extra validities I think we need to consider, and even within our core group, there's quite a bit of discussion and disagreement as to how to address or label these validities. But ultimately, I think it comes down to how well does an animal model mimic the human condition that you're trying to study. As well, I think we have far more complicated study designs, rightly or wrongly, within the preclinical world versus the clinical world. I've conducted preclinical systematic reviews where we included studies where they had 20 arms, which is absolutely bananas when you think about it. And I cannot think of a single clinical systematic review that I've conducted where we've come anywhere close to an experiment or a study where there's that many arms in a particular experiment. So a very, very big difference, I would say, and actually dealing with that is a big issue as well. In terms of our own efforts to develop PRISMA-Pre, we actually went back to Equator because they have a nice toolkit here on how to report guidelines or how to develop a reporting guideline. Again, I've just linked here with the QR code that goes right to this page on Equator. And when you look there, it's really interesting because they actually cite and heavily use this article, which was published back in 2010, written by David Moore, who's here in Ottawa. His office is right across the hall from mine, and then a few other well-known figures in the meta-research world. So they actually developed this. This was their expert opinion, and it's basically taken root as the way to develop reporting guidelines, rightly or wrongly. So for any of you that are thinking about developing any sort of guideline, I highly recommend you go to Equator. It has a nice set of tools, but also the caveat to all of this is that really it is based on expert opinion of these four folks. It's heavily weighted towards that. So what do they say and what did we follow? Basically, phase one, we had project leads and a core group formed and identified. Phase two, I told you all about this already. So we identified and characterized preclinical systematic reviews to understand the prevalence and reporting characteristics currently. We assessed current reporting and overall, like what did it look like and what was missing, and I've given you just a flavor of that from that study before. And then finally, what we're in the middle of right now is a Delphi process to identify essential reporting items. So the deliverables from all of this, we've registered this extension within the Equator network. That's something, again, for those of you that might be thinking about developing guidelines, is recommended. So if you register there, it basically provides a bit of a footprint. We've posted 3 protocols to the Open Science Framework as to what we're going to be doing within this research program. And of course, we published this cross-sectional study on pulse biology. In terms of our core group, lots of different figures from all around the world. I highlighted Sarah here from your center, and she's one of our leads. In addition, we have truly international representation, as you'll see here. And then some folks that I'm sure many of you are familiar with, Malcolm McLeod and Kimberly I already mentioned before. I should also mention that we have Lindsay, who is a medical librarian, who's an information specialist as well too. And of course, Emily and many people as well too. So lots of different folks from all around the world, and I think all well recognized for their expertise within preclinical systematic reviews. So what are we doing in this Delphi exercise? And I'll explain what Delphi is because I suspect some of you, especially some of the more junior folks, maybe are less familiar with this method. But Delphis, rightly or wrongly, are the method to develop and come to group consensus, especially around clinical guidelines. You'll see it very frequently, and also reporting guidelines. So our objective is to determine consensus on items that are essential to report in systematic reviews of preclinical, again, in vivo experiments. We have round 1, which has been completed. Basically, our core group voted within that. And what we did was we used the checklist that we used for our reporting assessment, and then we elaborated on that with a few other items that were brought up by core group members. In our next round, which is pretty much set to go, but excellent timing for this seminar because if you are interested, you can participate. Just send me an email. The requirements are, I believe, 2 systematic reviews that you've published, basically. So if you've published 2 preclinical systematic reviews, you're considered expert enough to definitely contribute to this because obviously you've come to understand what the process is around preclinical systematic reviews, and also have probably a very good understanding of what you should report. And at this point, you're probably basing reporting off of the PRISMA parent statement. So through Alex, I'll just put in the chat too. I'll just put my email there so you can actually reach out to me if you're interested. And then and I should mention that anyone who votes in round 2, you will be listed as a collaborator on the paper. So when you go to PubMed, for instance, there are the authors that are at the top, but you just press the button and all the collaborators actually pop up there. So it is actually listed and indexed as well too with collaborators. So that's the plan there, and we already have a journal we're going to go to with this, and they've agreed to help us work through that process with the indexing services. So after we get round 2 results, round 3, it'll just be the core, and that'll actually be combined with a consensus meeting. It'll be virtual. Everyone's so spread out, it's going to be impossible to bring people together with the lack of funding that we have for this project. So we will have a face-to-face virtual meeting, though, where we'll discuss and vote on any remaining items. So why would you want to do this? Well, basically, you want something that remains anonymous, and that helps prevent the loudest or more influential person from dominating consensus definition. So looking back at, I'm just going to flip back to the previous slide here, looking at this whole process where you have all these individuals, they're voting individually. No one's talking to each other. You're just at your computer voting on these items. And, I don't have Sarah beside me telling me what she thinks is the most important thing and trying to sway my vote one way or the other, right? So truly independent, or as independent as possible. Other than that, basically the third point that justifies this approach is that there is some evidence that a structured approach to group decisions are perhaps more accurate than from individuals. And that's not necessarily from guideline development, it's more from other fields where these methods actually developed from. In terms of the Delphi, what it looks like to folks that are voting in this, again, we start with a checklist of items. Participants rate them from 1 to 9, 1 to 3 being unessential, 7 to 9 being essential. Consensus is achieved when greater than 80% of participants rate in the same category. So already in round 1, which is completed, we've had several items that were rated as essential. So people voted between 7 to 9, and 80% of participants actually rated within that category. So they're actually going to be included in one form or another. Similarly, there are things that were deemed unessential. So again, 80% of participants voted 1 to 3, and those items have been kicked out of the checklist. There's an option for free text responses, and we got lots and lots of responses back from the first round, and that's actually made us redesign several aspects of the way we're actually presenting the questions as well too. So really great feedback, and I think our second round is a lot more refined because of that. And then for just interest's sake, I'm just mentioning that we are using survey-let software. We did quite a bit of research trying to figure out which software would be best for Delphi. There's several out there. There's flaws, fatal flaws, I would say, to all of them. So there are problems with the software too, but of the ones that we've looked at, again, I have no conflict of interest with them. We're paying them their usual rates, and it seems to be one of the best softwares. And I've participated myself in a number of different Delphis. And this seems to be one of the most streamlined. So in terms of unique elements, because you're probably wondering, well, what are the elements that we're thinking are unique? So I think number one, the title, clearly stating that you're using non-human or laboratory animal data is essential. Within our preclinical systematic review, our assessment, we found a number of studies which did not do that. It was just the fact that we're looking at the title and abstract that we recognized that there was animal data there. But just looking at the title alone, it was difficult to figure that out. And of course, when you're indexing and when you're trying to find articles, you want the title to be as accurate as possible. Another item, method for data presentation. So I already talked briefly about this issue, but multiple experimental arms. How did you actually deal with them in your reporting of what's happening? And also, how did you deal with them in terms of your analysis as well, too? Another issue that's become quite clear is that when you're talking about questions and how you frame them in systematic reviews, PICO is the acronym that many of us use, right, for clinical systematic review. So population, intervention, comparison, outcome. What we've suggested, and parts of this have reached consensus already, is that this population piece should probably be split apart into two elements when you're doing a preclinical systematic review of in vivo interventions. Number one, animals need to be discussed. So what animals were included, mouse, rat, non-human primates, whatever it might be, what did you include? And then what models were used, right? So I think very clear descriptions and sections on each of these are needed. And that's what's come through with some of our voting as well, too. Other items, intervention, comparison, outcome. There are some nuances there, which we basically largely just reframed a bit of the wording within the PRISMA statement to include the animal or the preclinical bit of it. But again, a lot of that actually carries through nicely from the parent PRISMA statement to the preclinical extension. Other unique elements, so external validity, I already mentioned this. A lot of discussion within our group as to what to label this. But again, what we're talking about is the applicability of each animal model, intervention comparator, and outcome to the human disease or the human health condition that we're actually interested in. So I think you can evaluate this in the multiple different ways. I said external validity here, but we're also can think about directness of evidence. You can think about translational validity or translatability. I know from a nice little editorial that was published from your center, Alex, that term translational validity was raised, and I thought there was some merit to that as well too. Within our own group, we like construct validity. I found very firmly through the voting that no one else likes it though. So we'll probably leave that off to the side. But again, I think there is a rich history to this term. Jonathan Kimmelman, who's doing a sabbatical right now at Quest, actually, he raised this as probably a nice term to think about some of these issues around external validity. It was in a paper that he published with the co-PI of my group, Dean Ferguson, in PLOS Medicine a few years ago now. Anyways, little plug for construct validity before it rides off into the sunset for this. But I think the other thing I want to plug here, I wasn't involved in this at all, but just so your people are aware, for those of you that are involved in Delphi's right now or planning to do a Delphi, this was just published in January. It's actually the first reporting guideline for consensus methods. Specifically, Delphi's is what they're talking about here. It was published in PLOS Medicine, and I've linked it here. Again, I wasn't involved in this, but it is now the reporting and the only reporting guideline around Delphi's, which was very odd because Delphi's, as many of you are probably aware, are so firmly entrenched in biomedicine. It's used in so many different aspects, whether it's clinical guidelines or reporting guidelines, as I've mentioned. But there was no actual reporting guideline for Delphi's before this. So really nice work there by this group. So that's it. Left some time for discussion here as well too, but take-home messages, I think you probably agree with me that preclinical systematic review reporting could be improved. There are unique challenges in preclinical systematic reviews that actually lack reporting guidelines or guidance at this time. We believe that preclinical PRISMA or PRISMA-Pre could improve reporting of preclinical systematic reviews. And our next steps are completion of the Delphi. I will leave my email in the chat here. So again, if you've completed a couple of systematic reviews, you are eligible to participate in the next round of voting. And I'm hoping groups like yours and others like CAMARADES and Circle will help promote the adoption worldwide of these reporting guidelines once they are published. They eventually will end up in PLOS Biology. We've already arranged for publication within that and potentially publication in more than one journal as well too. So I'll leave it at that, and we have plenty of time for discussion here as you see fit, Alex.